the resting phase, but these sources make it very clear it's anything but restful.
Yeah, resting is maybe misleading. Resting from division, maybe, but internally, it's incredibly busy. A whirlwind of activity, really. Interphase itself is broken down. G, S, and G.
GSG. Got it.
And importantly, before we even get to that GS, G Euro, M sequence, there's another state to consider, G, the sort of opt-out phase.
We'll come back to G. Let's focus on GEO Gap 1 first. If the cell cycle is like, say, a rocket launch prep, is GEO the initial systems check?
That's a good way to think about it. It's like the launch director's checklist. GS, all about growth. The cell gets bigger, it makes the proteins it'll need for the next stage, S phase, and it cranks out more organelles. If you're going to double everything and split, you need more hardware, right?
Makes sense.
But the most critical part of G, structurally, comes near the end. It's called the restriction point, or the GIA checkpoint.
Ah, the point of no return, the big decision moment.
Yeah.
What's the cell checking here? What's it looking for?
It's looking both outward and inward. Externally, are conditions right? Enough nutrients? Are the necessary growth factor signals coming in? Internally, though, the absolute top priority is the DNA. Is it damaged? Is it intact?
So it's checking the blueprint before committing to copying it.
Exactly. It's the big commitment point.
Hmm.
If conditions are good and the DNA is pristine, okay, green light for replication. But if it fails this check, well, the cell has options, but none are casual. It might pull out of the cycle into GIA,
The opt-out state we mentioned.
right? Or if the damage is too severe, it might trigger apoptosis-programmed cell death, which sounds drastic, but it's crucial protection for the organism against, say, potential cancer developing from a cell with faulty DNA.
Wow. Okay, so genetic fidelity is absolutely non-negotiable from the get-go. Once it passes that GEO checkpoint, we're into S phase. Synthesis. This sounds like the heavy lifting part.
It is replication central. This is where the entire genome gets duplicated. The key activity, making a faithful copy, turning each chromosome into a pair of identical sister chromatids joined at a spot called the centromere.
And the sheer scale of that is mind-boggling.
Yeah.
Billions of base pairs.
Copied with incredible accuracy, speed, and precision. That's the challenge.
So what's the S phase checkpoint looking for specifically? Is it different from the GEO check?
It shifts focus a bit. In GEO, it was, is the DNA okay to start copying? In S phase, it's more like, is the copying process itself going okay? Is it accurate? The machinery is constantly scanning. Are there errors being made by the DNA polymerases? Are any replication forks stalled?
Like quality control in the assembly line.
Exactly that. If it spots a problem, it can pause or slow down synthesis. Give the repair crews time to fix the mistake before the cell plows ahead with a potentially damaged genome.
Okay, replication complete. We move into GRO, gap 2, the final checks before the big M phase.
Right. GRO is about final preparations for the physical act of division. The cell really ramps up production of certain components. A key one mentioned in the sources is tubulin.
Tubulin. That's the building block for microtubules, isn't it?
Precisely. And microtubules are the core components of the mitotic spindle, this huge molecular machine that's going to pull the copied chromosomes apart. So GEO is largely about stocking up on tubulin.
And there's another checkpoint here, the GR checkpoint, last chance saloon before mitosis. We copied the DNA in S phase. What are we verifying now?
Two main things. One, is DNA replication 100% complete, no loose ends? Two, has any new DNA damage occurred since S phase finished? Or maybe damage that was missed earlier?
And this involves specific molecular players, right? The sources mention some key molecules involved in this verification.
Yes, absolutely. It's not just passively looking, it's an active surveillance system. If there is damage, specific sensor proteins detect it. Key players that often come up are kinases like ATM and ATR.
ATM and ATR, what do they do?
Think of them as the first responders. ATM, that's ataxia telangiectasia mutated, often detects double-strand breaks. ATR, ATM, and RAD3 related is more involved with single-strand issues or stalled replication forks. When they detect damage, they trigger signaling cascades.
So they sound the alarm.
They do. They phosphorylate, basically stick phosphate groups onto a whole bunch of downstream targets. And the ultimate effect of this cascade is to put the brakes on entering M phase. They inhibit the complex that normally triggers mitosis.
Like an emergency stop button activated by bad DNA data.
Exactly. And often working alongside these pathways or activated by them is another important protein, BRCA1. You might have heard of it in the context of breast cancer risk.
Yes.
Well, BRCA1 plays a critical role in DNA repair, especially repairing double-strand breaks through a process called homologous recombination. So if 8-MADR detect damage, they can activate BRCA1 to help fix it while simultaneously telling the cell cycle control system, hold up, don't enter mitosis yet.
So ATM, ATR, BRCA1, they're all part of this intricate damage detection and response network, making sure the cell doesn't divide with broken DNA.
Absolutely. They ensure the cell pauses until the DNA is repaired or trigger apoptosis if it's beyond repair.
Okay, before we finally jump into mitosis, let's just circle back quickly to GO, the state where cells aren't dividing.
Right, G, the quiescent state. It's like the cell is taking a break from the cycle. Some cells enter GO and stay there permanently. Think of mature neurons or cardiac muscle cells. They're terminally differentiated, they do their job, but they don't divide anymore.
So they exit the cycle for good.
Pretty much. But other cells can enter GO temporarily. Liver cells, for example, or lymphocytes, immune cells, they're just waiting for the right signal. If there's tissue damage or an infection, growth factors can call them back out of G and into GER to start dividing again. It's a facultative state for them.
Okay, got it. So G is either a permanent exit or a holding pattern. Now, the main event, M phase. Mitosis, the mechanical ballet.
This is it, where all that interphase prep pays off. Mitosis is specifically the division of the nucleus. It's incredibly dynamic, highly choreographed. Any mistake here?
Yeah.
Well, it can lead to daughter cells with the wrong number of chromosomes, which is usually disastrous.
And it happens in stages. The sources list 5, starting with prophase.
Prophase, condensation and setup. The first big visual change. The chromatin, which was diffuse and spread out during interphase.
Like spaghetti, you said.
Exactly. That spaghetti now condenses dramatically. It coils and folds up into distinct, visible structures, the chromosomes. This packing is vital for moving them around safely. You wouldn't try moving loose spaghetti across a room, right? You'd bundle it up.
Makes sense. And each chromosome at this point consists of the 2 sister chromatids copied in S phase, held together.
Yes, held together by protein rings called cohesin. We'll get back to those. While the chromosomes are condensing, another key thing is happening. The centrosomes start moving.
The microtubule organizing centers.
Right. They migrate towards opposite ends or poles of the cell. They're setting up the axis for division. And the sources highlight the structure here. Each centrosome has a pair of centrioles inside. And these centrioles have a very specific structure.
The 9x3 arrangement.
Yes, the 9x3 arrangement of microtubule triplets. 9 sets of 3 microtubules arranged in a cylinder.
That sounds incredibly specific. Why that structure? Is it for strength?
Strength, stability, and efficiency. It provides a really stable platform for nucleating or starting the growth of new microtubules. Remember, microtubules are made of tubulin dimers. In prophase, these dimers start polymerizing, growing outwards from the centrosomes. This is the beginning of the mitotic spindle.
So the 9x3 structure ensures a solid foundation for building this massive spindle machine.
Precisely. And radiating out from these centrosomes, you also get another set of microtubules called astral microtubules.
Astral, like stars.
Yeah, they radiate outwards like rays, but they don't generally attach to chromosomes. Instead, they reach out to the cell cortex, the inner surface of the plasma membrane. They act like anchors or guy ropes.
Stabilizing the whole spindle.
Exactly. They help position the spindle correctly within the cell and ensure the division happens along the right plane.
Okay, so chromosomes condense, spindle starts forming, centrosomes move apart. What's happening to the nucleus itself during prophase?
Well, the nucleolus, that dense spot inside where ribosomes are made, it usually fades and becomes indistinct. Ribosomal RNA transcription shuts down temporarily. But the major event concerning the nucleus is the breakdown of the nuclear envelope. It starts to fragment.
And this fragmentation, the sources mention, is quite clever. It doesn't just disintegrate randomly.
No, it's managed. The membranes break down into small vesicles. And importantly, these vesicles actually fuse with the membranes of the rough endoplasmic reticulum, the RER.
So the RER acts like a temporary storage depot for the nuclear membrane components.
That's a great way to put it. Holds onto them, ready for quick reassembly later in telophase. Very efficient.
Okay, prophase sets the stage. Next up, prometaphase, invasion and capture.
Right. Prometaphase kicks off when the nuclear envelope breakdown is complete. It's fully disassembled now, integrated into the RDR system. This means the spindle microtubules, which were forming outside the nucleus, can now invade the nuclear region.
The barrier is down.
The barrier is down, and the microtubules start hunting for chromosomes. Specifically, they're looking for the kinetochores.
Kinetochores, these are the docking stations on the chromosomes.
Exactly. Huge protein complexes built right onto the centromere region of each sister chromatid. They are the molecular handles that the microtubules need to grab onto.
So microtubules from the spindle find and attach to these kinetochores.
Yes, kinetochore microtubules attach. And as soon as they attach, movement begins. It's often described as a search and capture process. Microtubules grow and shrink, probing the space until they make contact with a kinetochore.
And this leads to that kind of tug-of-war we hear about, trying to line the chromosomes up.
It does. This is where motor proteins become absolutely critical. The cell needs to move the chromosomes and align them precisely at the cell's equator, an imaginary plane called a metaphase plate.
It's not just dragging them, it's positioning them.
Right. It's an active process, balancing forces. The key players here are motor proteins like kinesin and dynein, which use ATP to walk along microtubules.
Okay, let's break down their roles. Kinesin and dynein.
Generally speaking, kinesins tend to be plus-N directed motors. They walk towards the growing end of the microtubule, which is usually away from the centrosome. So kinesins associated with kinetochore microtubules often help push the chromosome towards the center, towards the metaphase plate. That's called anterograde movement.
Moving towards the equator.
Yes. Dynein, on the other hand, is typically a -N directed motor. It walks towards the microtubule organizing center, the centrosome pole. So dynein motors can pull the chromosome towards the pole, retrograde movement.
So the cell is using both pushing and pulling forces generated by these motors to get the chromosomes centered.
Exactly. It's a dynamic balance. And the crucial state it's trying to achieve for every single chromosome is biorientation.
Biorientation, meaning attached to microtubules from both poles.
Correct. One kinetochore on one sister chromatid is attached to microtubules from one pole, and the kinetochore on the other sister chromatid is attached to microtubules from the opposite pole.
And when that happens, tension builds up.
Yes. The pulling forces from opposite poles create tension across the centromere, stretching it slightly. This tension is actually a critical signal. It tells the cell, okay, this chromosome is correctly attached and ready for separation.
And the cell absolutely needs all chromosomes to be bioriented and under tension before it proceeds.
Absolutely non-negotiable. This is the job of the spindle assembly checkpoint, or SAC. It monitors the kinetochores. If even one kinetochore is unattached or attached incorrectly, say both sister kinetochores attached to the same pole, the SAC sends out a wait signal.
It puts the brakes on the whole process.
Completely halts the cycle until every single chromosome reports back, yep, I'm bioriented, under tension, ready to go.
Okay. Assuming the spindle checkpoint gives the all clear, we hit metaphase briefly where they're all perfectly aligned, and then boom, anaphase, separation.
Anaphase is dramatic and irreversible. The signal comes, the wait signal from the SAC is switched off, and that unleashes an enzyme called separase.
Separase! Its job sounds pretty clear.
It is. Separase targets those cohesin proteins, the molecular glue that's been holding the sister chromatids together all the way since S phase. Separase rapidly cleaves the cohesin rings.
Snaps the links.
Snaps the links. And instantly, the sister chromatids are no longer sisters. They become independent daughter chromosomes, and they start moving towards opposite poles.
How are they pulled apart? Is it just the microtubules reeling them in like fishing lines?
It's actually a bit more complex, a combination of two main things happening simultaneously. First, the kinetochore microtubules shorten. They depolymerize, primarily at the kinetochore end. So the track is literally being dismantled as the chromosome moves along it, effectively pulling it towards the pole. That's anaphase A.
Okay, pulling via shortening. What's the second mechanism?
The second mechanism involves the polar microtubules, the ones from opposite poles that overlap in the middle of the spindle. Motor proteins, specifically kinesins, sit in this overlap zone and actively push the microtubules apart. This forces the poles themselves to move further away from each other, increasing the distance between the separating sets of chromosomes. That's anaphase B.
Ah, so it's pulling the chromosomes towards the poles, anaphase A, and pushing the poles further apart, anaphase B.
Exactly. This dual mechanism ensures a rapid and efficient segregation of the two complete sets of chromosomes to opposite ends of the cell.
Okay, chromosomes have reached the poles. What's next? Telophase. Rebuilding.
Telophase is essentially the reverse of prophase and prometaphase. The segregation work is done. Now it's time to reestablish the two new nuclei. The chromosomes, having arrived at the poles, start to decondense, relaxing back into their more diffuse chromatin state.
And the nuclear envelope comes back.
Yes, using those components stored in the RER. Vesicles derived from the RER and containing those original nuclear membrane proteins gather around the clusters of chromosomes at each pole and fuse together to form new nuclear envelopes.
And the sources mention lamins being important here.
Critically important. Lamins are a type of intermediate filament protein that forms a meshwork lining the inside of the nuclear envelope, called the nuclear lamina. This provides structural support and is essential for the integrity of the reforming envelope.
Okay, nuclear envelopes reform. What else?
The nucleoli reappear. As the chromosomes decondense, the genes encoding ribosomal RNA become accessible again, transcription restarts, and the nucleoli assemble within the newly formed nuclei.
So by the end of telophase, we essentially have two distinct nuclei within the one still elongated cell.
Correct. Mitosis, the division of the nucleus, is complete, but the cell itself still needs to divide.
Which brings us to cytokinesis, division of the cytoplasm.
Right. Cytokinesis usually starts sometime during late anaphase or telophase and finishes shortly after telophase ends. It's the physical splitting of the cell into two daughters. And the mechanism, as the sources point out, is quite different depending on whether you're looking at an animal cell or a plant cell.
Because of the cell wall in plants, right?
Yeah, exactly. Animal cells, lacking a rigid wall, use contractile mechanism. A ring of actin microfilaments forms just under the plasma membrane, right at the former metaphase plate. Associated with this ring are myosin II motor proteins.
Actin and myosin, like in muscle contraction.
Similar principle. The myosin motors use ATP to pull on the actin filaments, causing the ring to constrict, like tightening a drawstring around the cell. This constriction creates the cleavage furrow, which deepens and deepens until it pinches the cell completely in two.
Okay, pinching in two for animal cells. What about plants with their rigid walls?
Pinching won't work there. Instead, they build a new wall from the inside out. Vesicles, filled with cell wall precursors and derived from the Golgi apparatus, travel along microtubules to the center of the cell, at the equator.
They line up in the middle.
They line up and start fusing together, forming a flattened, membrane-bound sac called the cell plate. This cell plate grows outwards, accumulating more vesicles and fusing with the existing plasma membrane and eventually the parent cell wall, until it completely separates the two daughter cells with a new cell wall between them.
Two very different solutions to the same problem of splitting the cytoplasm. It's amazing how evolution solved that.
It really is. Form follows function, dictated by the presence or absence of that wall.
Okay, that mechanical ballet is incredible, but the timing, the precision, it all hinges on the control system. Let's dive into that control tower now, the molecular regulation.
This is where it gets really elegant, I think. The whole cycle isn't just pushed by external signals. It's driven by an internal molecular clock. And the core components of this clock are two types of proteins: cyclin-dependent kinases, CDKs, and cyclins.
CDKs? Kinases phosphorylate things, right? They add phosphate groups to other proteins.
Exactly. CDKs are serothionine cornases. By phosphorylating specific target proteins, they act like molecular switches, turning those targets on or off, triggering events like DNA replication or entry into mitosis.
But they're cyclin-dependent. They don't work alone.
Nope, they're inactive on their own. They absolutely need to bind to a partner protein, a cyclin, to become active. And the key thing about cyclins is that their concentrations fluctuate throughout the cell cycle. They rise and fall in a predictable pattern.
Ah, so the CDKs are always there, but the cyclins come and go, turning the CDKs on and off at the right time.
Precisely. The cyclical appearance and disappearance of specific cyclins activate specific CDKs at specific phases, driving the transitions. It's this interplay that creates the rhythm, the internal clock.
Okay, so which cyclin-CDK pairs control which transitions? The sources mention specific partnership.
They do. It's like a relay race. To get past the restriction point in GO and commit to S phase, the key players are cyclin E binding to CDK2. Their activity phosphorylates targets needed to initiate DNA replication.
Cyclin ECDK2 for GO to S. Got it. What about S phase itself?
As cyclin E levels drop, cyclin A levels rise. Cyclin A partners mainly with CDK2 during S phase to help ensure replication continues smoothly and accurately. Then, as the cell moves towards G, cyclin A starts associating with CDK1.
Okay, so cyclin A spans S and G, and then the big one, entering mitosis.
That's the job of cyclin B. Cyclin B levels build up through G and peak at the GO transition. It partners with CDK1. This cyclin B CDK1 complex is often just called MCDK or MPF, maturation promoting factor.
MCDK, the master switch for mitosis.
It really is. When MCDK activity hits its peak, it phosphorylates a huge number of target proteins. This triggers chromatin condensation, nuclear envelope breakdown, spindle formation. Basically, it kicks off all the events of prophase.
It sounds like a one-way street once these CDKs get activated. But surely there needs to be a way to turn them off again and to make sure the cycle only goes forward.
Absolutely essential. You need irreversibility. The CDKs themselves can be regulated by other phosphorylation events, some activating, some inhibitory. But a major way the cycle progresses and ensures it doesn't go backwards is by destroying the cyclins themselves.
Destroying them. How does that work?
Through protein degradation, specifically the ubiquitin proteasome system. Think of it as tagging proteins for disposal. And an absolutely central player in controlling the exit from mitosis and ensuring irreversibility is a complex called the APCC.
APCC, anaphase-promoting complex cyclosome. We touched on this with the spindle checkpoint.
Yes, the spindle checkpoint controls the APCC. But the APCC itself is the executioner, in a way. It's a ubiquitin ligase.
Meaning it attaches ubiquitin tags to proteins.
Exactly. It attaches chains of ubiquitin to specific target proteins, marking them for destruction by the codisome, which is like the cell's garbage disposal unit. Once the spindle checkpoint is satisfied, remember, all chromosomes, bi-oriented and under tension, it removes the wait signal, allowing the APCC to become fully active.
And what does the active APCC target for destruction?
Two absolutely critical targets for getting out of mitosis. First, it targets cyclin B. Destroying cyclin B inactivates MCDK. And inactivating MCDK is necessary for the events of telophase to happen, things like nuclear envelope reformation and chromosome decondensation, which NCDK activity normally prevents.
Okay, destroying cyclin B lets telophase happen. What's the other target? You said it was crucial for anaphase itself.
Yes. The APCC also targets a protein called securin.
Securin. Is it securing something?
It is. Securin normally binds to and inhibits the enzyme, separates the enzyme that cuts cohesin.
Ah, so securin keeps the glue cutter inactive.
Exactly. So when the APCC gets activated, it ubiquitinates securin, targeting it for destruction. When securin is destroyed, separase is released and becomes active. Active separase then immediately cleaves the cohesin, holding the sister chromatids together.
And anaphase begins. The sisters separate.
Precisely. The activation of APCC and its targeting of securin and cyclin B are the key molecular events that make the metaphase to anaphase transition and the subsequent exit from mitosis sharp and irreversible.
Wow. So the cyclin CDK pairs are like the accelerator, pushing the cycle forward into the next phase.
And APCC is like slamming on the brakes for the previous phase's drivers, like cyclin B, while simultaneously cutting the ropes via securin separase to let the next stage proceed irreversibly.
That's a really clear picture. It's this beautiful coordination between the biochemical timing, the rise and fall of cyclins, the activation of kinases, the targeted destruction, and the physical mechanics, the microtubules, motors, actin filaments.
And it's incredibly energy intensive. Let's not forget the fuel source.
Right, ATP. Where is it primarily used?
Everywhere. DNA replication in S phase consumes vast amounts of ATP to build the new strands. Building the spindle requires ATP for tubulin polymerization. Then the motor proteins, kinesins, dyneins, myosins, they all hydrolyze ATP to generate force and movement. Shortening microtubules also requires energy. It's a high-energy operation.
So we have these nanoscale machines, 25-nanometer microtubules, tiny actin filaments, motor proteins walking along them, all orchestrated by this incredibly precise molecular clock fueled by ATP.
And anchored by things like the intermediate filaments, the lamins, providing structural support for things like the nucleus. The feedback loops are crucial too. High cyclin B drives mitosis, but mitotic events activate APCC, which destroys cyclin B, ensuring the exit from mitosis. It's self-regulating.
Okay, we have a solid grasp of mitosis now. But cells divide in another way too, don't they? For sexual reproduction. Let's compare mitosis with meiosis.
Yes, a very important comparison. Mitosis, as we've discussed, is for growth, repair, asexual reproduction in some organisms. The goal is identity. You start with one diploid cell, 2ken, meaning two sets of chromosomes, and end with two genetically identical diploid daughter cells. 2n is 2n, one division.
Same number of chromosomes, identical genetic information.
Exactly. Meiosis, however, is exclusively for producing gametes, sperm, and eggs for sexual reproduction. The goal isn't identity, it's diversity and reducing the chromosome number.
So how does it achieve that?
It involves 2 rounds of division, meiosis the first and meiosis the second, and you start with one diploid cell, 2n, but end up with 4 genetically non-identical haploid cells. N, meaning one set of chromosomes, 2nn.
4 non-identical haploid cells. How does the non-identical part happen? And the reduction in chromosome number?
The key differences happen in meiosis the first, particularly in prophase I. In mitotic prophase, chromosomes just condense. But in meiotic prophase I, something much more complex happens. First, homologous chromosomes, the pair of chromosomes carrying the same genes, one inherited from each parent, find each other and pair up precisely along their length. This pairing is called synapsis.
Okay, homologous pairs stick together.
And then, while they're paired up, they undergo crossing over. Segments of DNA are literally exchanged between the non-sister chromatids of the homologous pair.
Swapping genetic material between the maternal and paternal chromosomes.
Exactly. This shuffles the genetic deck, creating new combinations of alleles on each chromatid. This is a major source of genetic variation, and it simply doesn't happen in mitosis.
Okay, crossing over in prophase I, what else is different in meiosis of first?
The way they line up in metaphase 4. In mitosis, individual chromosomes, each with 2 chromatids, line up in the metaphase plate. In metaphase of meiosis, it's the homologous pairs, each pair consisting of 2 chromosomes, so 4 chromatids total, called a bivalent or tetrad, that line up together at the plate.
So pairs line up, not individual replicated chromosomes.
Right. And this dictates what separates an anaphase first. Mitotic anaphase, sister chromatids separate. But in anaphase of first of meiosis, the homologous chromosomes separate and move to opposite poles. The sister chromatids remain attached to each other.
Ah, so meiosis lyse separates the homologous pairs, reducing the chromosome set number from diploid to haploid right there.
Precisely. The cell divides, cytokinesis the first, resulting in 2 haploid cells, although each chromosome still consists of 2 sister chromatids. Then, these 2 cells immediately enter meiosis the second.
Meiosis the second.
Meiosis the second is mechanically much more like mitosis. Chromosomes line up individually at the metaphase to a plate, and in anaphase the second, the sister chromatids finally separate. This results in a total of 4 haploid daughter cells, each genetically distinct due to crossing over and the random assortment of homologous chromosomes in meiosis the first.
That distinction is crucial. Mitosis for identical copies, meiosis for having chromosome number and creating variation.
2 fundamental processes with very different outcomes, driven by modifications to the basic division machinery.
Now, inevitably, when you have such a complex, tightly regulated system, things can go wrong. And when cell cycle control breaks down, that leads us directly to cancer, doesn't it?
Fundamentally, yes. Cancer is, at its core, a disease of uncontrolled cell proliferation, which means it's a disease of a broken cell cycle clock. The mutations involved typically do one of 2 things. They either jam the accelerator pedal down, or they cut the brake lines.
Let's talk about the accelerator first, oncogenes.
Right. Oncogenes arise from normal genes called proto-oncogenes. Proto-oncogenes usually promote cell growth and division, but only when the cell receives the proper signals. Mutations can convert these proto-oncogenes into oncogenes, which are permanently switched on. This is a gain of function.
So they constantly signal divide, 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 even without external growth factors.
Exactly. They provide constitutive signaling. Classic examples are mutations in genes like RAS, which is part of a key signaling pathway, relaying external growth signals, or MAC, a transcription factor that turns on genes involved in proliferation. Mutated versions just keep driving the cycle forward relentlessly.
Okay, that's the stuck accelerator. What about cutting the brake lines? Tumor suppressor genes.
These are the protective genes. Their normal job is to halt the cell cycle if something is wrong, DNA damage, lack of nutrients, etc., or to trigger apoptosis if the problem is irreparable. Cancer often involves loss-of-function mutations in tumor suppressor genes. The brakes fail.
And the 2 classic examples mentioned everywhere are P53 and RBA.
Absolutely central. We talked about P53 as the guardian of the genome, crucial for the GO checkpoint. If you lose functional P53, cells with damaged DNA can sail right through the checkpoint and into S phase, accumulating more mutations.
And RB, the retinoblastoma protein, how does it act as a brake?
RB's main job is to control entry into S phase by regulating a group of transcription factors called E2F. Normally, RB binds to E2F and keeps it inactive. When the cell receives signals to divide, passing the GAT checkpoint, cyclin ECDK2 phosphorylates RB. This phosphorylation changes RB's shape, causing it to release E2F.
And free E2F turns on genes needed for S phase.
Exactly. Genes for DNA polymerase, enzymes for nucleotide synthesis, etc. Now, if RB itself is mutated or missing, it can't hold on to E2F. So E2F is always active, constantly turning on S-phase genes, leading to uncontrolled entry into the replication cycle. Loss of RB is like removing a key gatekeeper at the GS transition.
So, oncogenes push, failed tumor suppressors release the brakes. But cancer cells often acquire other tricks too, like becoming immortal.
Yes, they overcome the natural limit on cell divisions. Normal cells have telomeres, protective caps at the ends of chromosomes, which shorten with each replication cycle. Eventually, they get too short, signaling the cell to stop dividing. This is senescence, the Hayflick limit.
A built-in lifespan counter.
Right. Many cancer cells manage to reactivate an enzyme called telomerase. Telomerase can rebuild the telomere ends, effectively stopping the counter and allowing the cells to divide indefinitely, achieving replicative immortality.
And they need fuel and oxygen for all this division. They need a blood supply.
They do. As a tumor grows, the cells in the center become starved of oxygen and nutrients. This hypoxic environment triggers them to release signaling molecules, the most famous being VEGF, vascular endothelial growth factor.
VEGF signals for new blood vessels.
Exactly. It induces angiogenesis, the growth of new blood vessels into the tumor mass. This provides the supply lines needed to sustain the uncontrolled growth. It's a crucial step for a tumor to grow beyond a tiny, localized clump.
These mechanisms aren't just abstract concepts. The sources give some really stark clinical examples of what happens when these specific pathways fail.
They do. Take Li-Fraumeni syndrome. It's a rare inherited disorder where individuals inherit one faulty copy of the TP53 gene.
So they're born with one p53 break already partially disabled.
Essentially, yes. And because p53 is so central to preventing damaged cells from dividing, these individuals have a dramatically increased risk of developing multiple types of cancer, often at a young age. It starkly illustrates p53's role as a master tumor suppressor.
And external factors can sabotage these systems too, like viruses.
A prime example is HPV, the human papillomavirus, particularly high-risk strains linked to cervical and other cancers. HPV produces viral proteins that directly target and disable the cell's tumor suppressors.
How does it do that?
It makes two key oncoproteins, E6 and E7. E6 targets p53 for destruction, essentially eliminating the geocheckpoint guardian. E7 binds to and inactivates the RB protein, removing the E2F gatekeeper.
Wow, a double hit, taking out both p53 and RB.
Exactly. By neutralizing both major geobreaks, HPV hijacks the cell's machinery, forcing it to divide repeatedly, which creates more opportunities for cancer-causing mutations to accumulate.
It's fascinating and scary how these pathways can be disrupted. And it's not just cancer, is it? The sources mention a link to Alzheimer's disease.
Yes, this is a really interesting, perhaps less intuitive connection. Neurons are normally postmitotic. They're in a permanent G0 state. But studies on brains affected by Alzheimer's have found evidence that some neurons aberrantly try to re-enter the cell cycle.
They try to divide when they shouldn't.
They try to initiate the cycle, yes, but neurons aren't equipped to complete mitosis properly. This aberrant re-entry seems to be highly toxic to the neuron, triggering signaling pathways that lead to apoptosis cell death. So inappropriate cell cycle activation in neurons might actually contribute to the neurodegeneration seen in Alzheimer's.
That's a surprising link, but it also highlights the potential flip side. If we understand the off switch so well, maybe we can learn to turn it on controllably for therapeutic purposes. Regenerative medicine.
That's the huge hope, isn't it? If breakdown causes disease, maybe precise control enables repair. Imagine being able to coax cells out of GO, guide them through a limited number of safe divisions to, say, regenerate damaged heart tissue or repair spinal cord injuries, and then reliably signal them to stop and differentiate or return to quiescence.
Mastering the checkpoints for controlled proliferation.
Exactly. Understanding the molecular signals, the specific cyclin CDK activities, the roles of p53, RB, the growth factor pathways, gives us the potential toolkit to manipulate cell division for therapeutic benefit. It's a major goal of regenerative medicine.
Which leads us naturally to how we currently interfere with the cell cycle, primarily in cancer treatment. Chemotherapy often targets dividing cells, right?
Yes, because cancer cells are, by definition, dividing rapidly and often uncontrollably. They are generally more susceptible to drugs that interfere with the division process than most normal quiescent cells. Many classic chemo drugs exploit the mechanics of M phase.
Like targeting the microtubules of the spindle.
Precisely. There are two main strategies here. One is to overstabilize the microtubules. Drugs like Taxol or paclitaxel do this. They bind to microtubules and prevent them from depolymerizing, from breaking down.
Why is that bad? Isn't stability good?
The spindle needs to be dynamic. Microtubules have to grow and shrink to capture chromosomes, align them, and then pull them apart in anaphase. If Taxol freezes the microtubules, the spindle gets locked. The cell arrests in mitosis, can't complete division, and usually undergoes apoptosis.
So, too much stability is just as bad as too little. What's the opposite strategy?
Preventing microtubule formation in the first place. Drugs like vincristine and vinblastine, derived from the periwinkle plant, bind directly to the free tubulin dimers and inhibit their polymerization into microtubules.
So, no spindle can form properly.
Right. Again, the result is mitotic arrest and cell death. So you can kill dividing cells by either preventing spindle breakdown, Taxol, or preventing spindle formation, vinca alkaloids.
Beyond hitting the structure, can we attack the DNA itself during the cycle?
Absolutely. That's another classic chemotherapy approach. Drugs like cisplatin, are DNA-damaging agents. They cause cross-links within and between DNA strands.
Creating massive errors for the checkpoints to find.
Exactly. In rapidly dividing cells, the DNA damage burden from cisplatin becomes overwhelming. The GO checkpoint, or sometimes even S-phase checkpoints, detect this irreparable damage and trigger apoptosis.
And more recently, we have drugs that target the control system itself, right? The molecular clock components.
Yes, this is the era of targeted therapy. Instead of general poisons, we're developing drugs aimed at specific molecular drivers. A key class here is CDK inhibitors.
Blocking the engine components directly.
Precisely. For example, drugs like palbociclib are inhibitors of CDK4 and CDK6. These CDKs, when partnered with cyclin D, are important early in GO for phosphorylating RB. By inhibiting CDK46, these drugs prevent RB phosphorylation.
So RB stays active, holding on to E2F.
Correct. E2F remains inhibited, the genes needed for S-phase aren't turned on, and the cell cycle arrests right there at the GO checkpoint. It stops the proliferation before DNA replication even begins. This is particularly effective in certain types of breast cancer, where this pathway is overactive.
It's incredible. From understanding the basic mechanics like the 9x3 structure of centrioles to targeting specific kinases like CDK46 with drugs, the journey has been huge.
It really has, and I think the key takeaway for you, the listener, is this idea of balance. The cell cycle is this constant tightrope walk between the drive to replicate and the absolute necessity of restraint and control.
You need the physical machinery, the cytoskeleton, the motors, all fueled by ATP, but it's utterly useless or dangerous without the precise timing dictated by the cyclin CDK clock and the crucial safety net of the checkpoints and destruction pathways like the APCC.
And those safeguards, the tumor suppressors like P53 and RB, are just paramount. Losing them is often that first critical step towards uncontrolled division in cancer. And the details from the sources like the RER, recyclonuclear membrane, or the dual push-pull forces in anaphase just highlight how optimized and elegant this whole process is.
It really drives home the tension, doesn't it? Dysregulation leads to cancer, but perfect regulation, that holds the key to regeneration.
Exactly. The same machinery, the same clock. It's all about whether it's running correctly or running wild.
Which leaves us with a really provocative thought, building on that regenerative potential you mentioned. If we truly understand the molecular intricacies now, the specific CDK partners, the P53 damage response network, how the APCC triggers irreversible transitions, the signals for G entry and exit, does this mean that achieving total programmed control over this molecular clock is the ultimate prize? Could mastering it be the gateway not just to better cancer therapies, but to actually realizing that dream of regenerative medicine, the power to safely switch cells on to divide and repair tissues, and then reliably switch them off again?
It certainly feels like the ultimate goal in controlling cell fate. True biological mastery might just boil down to having that precise on-off switch for the cell cycle deployable exactly when and where it's needed.
A powerful idea to consider. That concludes our deep dive into the cell cycle's molecular clock and cellular